and welcome to Cooking with Science. In today's segment, we will be learning about DNA, and we will also be making our very own strand of DNA. But before we begin, we will need to learn more about this special acid and what its function is. Let's roll the clip. According to Citable 2014, deoxyribonucleic acid, more commonly known as DNA, is a complex molecule that contains all of the information necessary to build and maintain an organism. Each gene contains an acid called deoxyribonucleic acid. Whenever organisms reproduce, a proportion of their DNA is passed to their offspring. All the cells found in an organism exclusive of the gametes contain the same DNA. The functions of DNA is vital for inheritance and the coding of proteins. DNA transfers genetic instructions to different cells in the body. When cells reproduce by mitosis, the DNA is replicated identically. The strands of DNA are unzipped and an exact copy is made by matching each base pair with its complementary base. Ribosomes read the RNA code one letter at a time and draw amino acids from their surroundings and attach them together in a chain according to the RNA code. Every 3 letters of the RNA code inform the ribosome which 3 of the 20 amino acids should be added next. The amino acids join together to form polypeptide, chains of amino acids. The polypeptides determine characteristics and phenotypes, such as blonde hair and blue eyes. Proteins are made up of one or more polypeptide. Errors in DNA replication can lead to mutations and diseases such as sickle cell anemia. DNA was discovered in the early 1950s by James Watson and Francis Crick. Now that we know more about DNA, let's make our very own strand of DNA. The structure of DNA is shaped as a double helix with 2 strands twisted together. Firstly, let's add alternating units of sugar and phosphate. These components make the uprights of the DNA and are known as the sugar-phosphate chain. Secondly, we will be adding molecules called nitrogen bases. These are located in the middle of the sugar-phosphate chain. There are 4 different nitrogen bases. 2 complementary bases join together to form a base pair. 1,000 base pairs form one gene and 3 bases join together to form a codon. The 4 nitrogen bases are adenine, thymine, guanine, and cytosine, and they are expressed by the letters A, T, G, C. Adenine and guanine are both purine bases. In comparison, thymine and cytosine are prenamine bases. The chemical structure allows for only 2 nitrogen bases to join together to form a base pair. The complementary base pairs are adenine and thymine, and cytosine and guanine. Hydrogen bonds bound together the 2 complementary base pairs, also known as nucleotides. The sequence of base pairs carry the genetic information required for protein synthesis. The order of base pairs determine how the amino acids join to form a protein molecule. Now that we have added all the elements that a single strand of DNA is comprised of, let's take a look at the final product. Thank you for watching. And always remember, think like a proton and stay positive.